Thank you very much, and thanks for inviting us. I don't think I have to tell anybody about ischemia, reperfusion, and heart disease here. We've been talking about it all morning. It's one of the major events, one of the causes of death worldwide. We've heard a lot of mechanisms involved. However, we and others have shown that alterations in mitochondrial structure and function play a significant role. I'm not sure if I have there I do. Okay. Here you can see in a control electron microscopy, the mitochondria are electron dense, they're round, versus in ischemia, this is 30 minutes of a normal thermic ischemia, they become very enlarged, they lose their function, calcium accumulates, those little black dots. You lose enzyme activity, which means you've got less ATP and energy generated, and you end up with a lot of mitochondrial DNA damage. This results in changes in the mitochondrial proteome and the transcriptics and leads to cell death pathways. All of these events occur during ischemia, and despite reperfusion, they persist, and they end up with a decreased cell viability and decreased function. Now, throughout the years, there's been many approaches to try and salvage mitochondria. These include various different pathways and functions. All of them have dealt with just a small part of the pathway, and they've essentially all been shown to be ineffective clinically. About 15 years ago, we had been looking at this, and we decided that perhaps another way to look at this is rather than look at a small part of the mechanism or a small part of the pathway, would be to treat the organelle itself. So in this way, we would replace the damaged mitochondria in the heart with mitochondria that were viable that we had isolated from another part of the body. To do this, we've developed an isolation procedure. What we do is we extract a small piece of tissue. Here they are up at the top. We extract them with number 6 biopsy punch. In the adult population in which we were doing a mini thoracotomy, they would be extracted from the pectoralis major. In a pediatric population where there's a sternotomy, we do it from the rectus muscle. We only need one of those pieces of tissue. We take 2 because some of our babies are so sick. In order to find a viable piece of muscle tissue, you actually have to dissect both pieces to get a tissue to do it. The tissue is then homogenized rapidly. It's by a mechanical homogenization, which is on a computer program, so it occurs easily. It's then digested with a generally accepted as safe protease, and then it's filtered through a series of filters, and the product is centrifuged down and kept. And the whole process takes less than 30 minutes, and we do it right at the bedside. The mitochondria coming out are viable. On the top you can see there's a bright field shot of the mitochondria. The middle one is mitotracker red, showing mitochondrial viability and mitochondrial potential. The overlay is the merge. We get a product from that one piece of tissue. We get about 1 times 10 to the 9th to 1 times 10 to the 10th mitochondria. The mitochondria that we get out are 99.9% pure. Here's an EM of them. You can see that we have one little star on there. This is an EM shot. The way we centrifuge them down, that could actually be the end of a mitochondria, but we decided to count that as a contaminant. All the mitochondria maintain complex function. They're able to function. They use oxygen, and they produce ATP. So this is important because the advantages of this are it's obtained from the patient. The tissue is obtained from the patient, so it's autologous. The isolation procedure is rapid. It's less than 30 minutes. We get intact, viable mitochondria from this. We found out earlier on that if we take mitochondrial DNA, old mitochondria or mitochondria don't work, mitochondrial RNA, the proteins or the complexes, or just exogenous ATP, it doesn't provide for cardioprotection. You need intact, respiration-competent, viable mitochondria. So as I said, it can be performed at the bedside, and we regularly do this in the ICU unit or in the surgery. And it can be performed as needed within the same operation because it takes so little time. And it allows the surgeon, therefore, to make a decision, see what they might need. The small pieces of tissue we can grab at any point in time, and it's ready once that decision has been made. Now, the way we've been doing it is we've been directly injecting the mitochondria into the tissue. Here we can see their mitochondria are labeled with F18 rhodamine and the iron oxide. You can see by the PET image, the mitochondria by direct injection go just into a single area. That's verified with the MRI. Here you have the micro CT and the PET image combined with the merged one. They tend to go into just one area. You can also inject them in by the vasculature. Here you have them in the lower frame. You can see them once again by PET, micro CT going around. That white line here is just to show, that's stainless steel suture, just to show where we tied off the LAD for the regional ischemic model we had. The mitochondria go into both viable and ischemic tissue. They go in a little bit higher into the ischemic tissue, as you can see. We inject for direct injection, we just use a tuberculin syringe, 28, 32 gauge needle. As I said earlier on, it's localized delivery, simple, safe. We normally use multiple injections, about 100 microliters each one. There's no flush back. Each one we put in about 1 × 10⁸ mitochondria. The only drawback on this is for posterior application, you have to manipulate the heart. The other way is to do direct injection via into the coronary ostium. We normally do this as a carotid cut down. You use a 5S catheter. It's simple and safe. There's no effect on coronary patency. We've tested this out on pig hearts using ADH and or epinephrine, and there's no change in the ECG flow rate or on angiography. So usually we do one injection of 5 mils with a 5 mil flush. It goes, as I said, it goes access to the whole heart, but it only goes to the heart. You'd think that if you injected mitochondria into the coronary ostium, especially in a diseased heart, you would get that flushed out and you would go into the various other organs. Here it is. This is actually in a rat that we first did it. You can see it's only in the heart itself. And here is in a pig. This is just studies we've just done. This is a pig that's used, we use 6 times our regular dose. This is actually a mistake by my surgical fellow. She thought the 6 doses I gave her were going to be put in all at one time and just flushed in 30 mils all at one time rather than 5 mils, 5 mils, 5 mils. What you can see here is up at the top, that's the syringe and the catheter that we had to leave in place. We use the pediatric MRI, and they tend to frown upon us leaking pig stuff all over. So as you can see, it just goes directly into the heart, doesn't go anywhere else. Once they're delivered, the mitochondria are rapidly taken up by various cells in the myocardium, and in fact, in every organ that we've looked at. We've demonstrated this using mitochondrial fluorescence probes. We've done compartmental fluorescence. Over here, we've done xenogenic mitochondrial transplantation using human mitochondria into either a pig or a rabbit heart. The human mitochondria are used to denote that where they are, because obviously if we used autologous mitochondria, we wouldn't be able to distinguish one from the other. The human mitochondrial, there's an antibody that only reacts to human mitochondria, so we're going to be able to tell that out. You saw the F18 and iron oxide go right into the cells. This is an interesting concept because we can put a 30 nanometer iron oxide particle on these mitochondria. They go into the cells, so this would allow for eventual drug delivery to anything that we want in addition. We've also shown by immunogold, and lately we've been doing A lot of studies using high-resolution microscopy where we've labeled the mitochondria with RF, red fluorescent protein or a green fluorescent protein using a baculovirus vector. This is actually done by a friend of mine, a colleague who enjoys sitting in dark rooms a lot more than I do. This is into a human iPS cardiomyocyte, and you can see the red and green. The green mitochondria are from a human cardiac fibroblast. Those are the human cardiac fibroblast mitochondria in there. The way they go in is through, and this is Dr. Cowan's work, they go in by endocytosis. We had done this earlier on looking at just blockers. They quickly come in. Here it is. The green mitochondria are over here. I'm going to stop this and so we don't go into an epileptic fit on the audience. But you can see the green mitochondria come in. They come in very rapidly. They come in and we can see them by, they're in earlier with pre-endosomes. Then they go and released. Then they're late endosomes and released. And then they go off and they fuse with the mitochondria within the cell. We've done calculations on this, and our early calculations showed that we thought we were only getting in 5% of the mitochondria that we put into the cells, but in fact what was happening is, as you can see, the signal gets quenched once they start fusing with the other mitochondria. So we were underestimating how many mitochondria we're getting in the cell. So the shortest time period we can do right now is 2.5 minutes, and at 2.5 minutes we got approximately 48% of the mitochondria inside the cells. The others are outside. Now, we've tested the use of autologous mitochondrial transplantation in a series of animal studies. They've been mostly large animal studies. It's the same model that we've used all the time. This is a model we've been using for the last 30 years. It's a regional ischemic model. We snare the LAD temporarily, do 30 minutes of ischemia, warm glomerular ischemia, regional ischemia. At the end of 30 minutes, we either deliver the mitochondria or we deliver the vehicle that the mitochondria are in, and then we allow reperfusion for 2 hours or 28 days. 28 days is as far as we've gone with the animal models because, quite frankly, I have to rent them a room here. It would be cheaper than keeping them up any longer. The injection of the mitochondria is totally safe. We have no arrhythmogenicity. We've checked this out with ECG, QRS stimulation. There's no change in the corrected QT level. We've done this with optical mapping where we've actually injected in 5 times what we normally inject into any animal. There's no change in the electronic signal. Signal propagation. There's no inflammatory response with them. HSCRP, high-sensitive CRP, all the inflammatory markers are all down. Nothing is there whatsoever. There's no autoimmune response. We've collaborated with the people over at Mass General to check this out, thinking because we put in a lot of mitochondria, it could be possibly you get an autoimmune response. There's nothing there. We also looked at immune response. There's no direct, there's no indirect, acute, chronic, alloreactivity or allorecognition. And there's also no DAMPS response. The mechanism of action is a little bit, it involves a lot of different mechanisms on the mitochondria. You know, for a long time we used to get mitochondria the powerhouse of the cell, and quite frankly when I hear that I sort of cringe because as we keep going on every day we find something new is involved with these mitochondria. They act independent of calcium signaling or ROS signaling. Of course, you have to remember that we've extracted them from the tissue. They are no longer hooked up to the connective tissue inside the cell. They're no longer lined up, so they have no calcium signaling coming into them. They don't act as a calcium sink. They don't act as a ROS sink. Neither do they give off ROS. What they do, though, is they increase the intercellular total ATP. So you get a huge increase in ATP, and you increase the oxygen consumption and uptake in the cellular respiration. They also change around the proteomic pathways, and this occurs rapidly. We see this in 10 to 15 minutes after we inject them. This is the same time period where we see the change in epicardial ultrasound changes within the heart. Here's just a heat map. You can, the papers are referenced over there, but the first two lanes are just control hearts that had no ischemia whatsoever. The next ones are from vehicle, and then the last three are from mitochondria, and you can see the changes in those. We also get cytokines changes, and it replaces the damaged mitochondria as you wish. All of this leads to increased cardiomyocyte viability and increased post-ischemic function. Here's just to show you the echocardiograms on that. So what I hope I've been able to show you here is that this autologous mitochondrial transplantation, I would hope, adds to the armamentarium of any surgeon that we have here. Isolation process is rapid, it's easy, it's safe, can be performed at the bedside. The specificity of distribution is right there. We can do it by both ways. It's robust, lasts 28 days, and it seems to be effective. We've now gone a first in human, and my colleague, Dr. Mani, will show you that later. Thank you very much.